Thanks very much, Ali, and thanks to the organizers. I think I'm last in this group because at Tempus we deal mostly with patients that have cancer, patients that have metastatic cancer. So we represent kind of the purple bar of one of Gabe's early slides. And in a way, our goal at Tempus is to make that overall survival higher than it is. And I think one of the ways we're doing that is looking at the combination of clinical data with genomic data, and that's really going to be the theme of the talk. I think when we talk about personalized medicine, the genomic revolution, I think its potential role in cancer is really the poster child for that, and especially metastatic cancer, matching alterations with targeted drugs, leading to better outcomes, hopefully, that sort of thing. But even though there's been a lot of press, there's also been some negative press, I think, recently. Maybe it's been overhyped. Are we really getting better results by doing broad panel next-gen sequencing, for example, matching them with targeted therapies? I think, like most new technologies, there's always a period where the hype is probably overblown, and then it kind of settles down to reality. But I also think there's no doubt that the promise of the genomic insights that we're making with cancer now are really tremendous. One of the reasons this is so important, and I love this slide, the percent of patient population for which a particular drug is ineffective. Well, look at cancer. Taking chemotherapy, for example, 20%, 25% is a good response rate. So we know, as a medical oncologist, giving chemotherapy to a patient, 3 out of 4 times it's not going to work. You know that when you give it. Of course, the problem is you can't tell which is the one of the 4 in which it may work. And when it does work, it's certainly not curative in most metastatic situations. So this, of course, is one of the values of finding a target, finding an EGFR mutation in a lung cancer. Then you can have a particular drug attacking that target, and your response rates here can go up 50%, 60%, still not 100%. And there are side effects, obviously, with even targeted drugs. The whole goal here, see if I can use the pointer, is to get away from just histologic-based diagnoses, as we're used to seeing breast cancer, prostate cancer, lung cancer, and find through molecular diagnostic testing those genetic or genomic similarities among the different cancers and treat them the same way. That's really the principle of genomics in metastatic cancer. As you start breaking these down, though, some of these alterations are rare. You break them down into certain tumor types are even rarer, and it raises the question, are these targeted therapies transferable across tumor types? We like to think they are. We like to think that an anti-EGFR mutation drug is going to work regardless of the tumor type, but we know, in fact, that that's not the case. So the problem is, with all of the different potential targets, copy number changes, insertions, deletions, various mutations, rearrangements, and so forth, you have a large number of potential targets, obviously a large number of tumor types. And when you start putting them together, you get pretty small data sets on which to look at the efficacy of certain drugs. Here's a good example that I saw in one of our reports at Tempus just last week. We've all heard about checkpoint inhibitors. Doctors routinely stain now for PD-L1 by immunohistochemistry. They'll use one of the checkpoint inhibitors, even though the criteria are still a little bit loose. Well, what do you do with a patient like this, where we found a PD-L1 copy number gain? Well, you could argue this is going to lead to overproduction of the PD-L1 protein. The patient could be IHC positive, though it isn't necessarily the case. What's the data to show that a PD-L1 copy number gain patient is going to respond to a checkpoint inhibitor? Well, if you look, as we do, we scan the literature, and it turns out this has been looked at. In this particular patient, a study of 118,000 cases, PD-L1 amplifications were seen in 843, including 100 different types of cancer. They were not really associated with any particular tumor mutational burden. they didn't always correlate with immunohistochemistry. So you can't say, well, do an immunohistochemistry test. If it's positive, treat. If it isn't, don't treat. And there are a lot of particular reasons for this, but the point is there was not correlation. So a doctor gets that report and they say, should I use a checkpoint inhibitor or not? Turns out that there really is very little literature on that. In fact, if you look at the Cancer Genome Atlas and you look at the number of different mutations in certain tumor types, and you're down to a handful of cases. What's even more problematic is that there is no clinical follow-up information on these cases. If one of these patients did get a checkpoint inhibitor, let's say, in that PD-L1 amplified case, there would be no way to find it because the clinical information generally isn't recorded. As we know, only 5% of patients in this country are on clinical trials where you record the data. So another problem is resistance. Does the drug work in the first place? If it does work, how does the body become resistant? Here's a fairly graphic example, but this is a case of a 38-year-old man with melanoma that happened to have a BRAF mutation. And we know that vemurafenib is one of the really poster child drugs in BRAF mutated melanoma. And sure enough, the patient responded within a short period of time very well, as you see. But very typically, these patients recur almost as fast as they've responded. So what is it about this patient's mutational profile that maybe predicted a response but also a relapse? Again, if you don't collect the fact that this patient relapsed quickly, you're going to lose that information. So I think it's very, very important with the complexity of all of these pathways, not only to do the genomic analysis, but to collect the clinical information. And this, I think, is what Tempus's mission is to a large degree. We're trying to take the 95% of metastatic cancer patients who are not on clinical trials and get as much clinical information from them as we can, those that are sequenced. And this is a very challenging issue, as you can probably imagine. Doctors use different medical record systems. The medical record systems don't talk to each other. Even here in California, Kaiser, which uses Epic, uses different versions of Epic, and I've told that the Kaiser centers don't talk to each other. So it's a really challenging issue. But that's really, I think, what Tempus is all about, to try to get as much genomic information as we can, but then to match it with the clinical outcome information to make some sense out as to what the genomic changes really means. We're a relatively new company. We're about 3 years old, based in Chicago. We're up to about 600 employees now, and we like to think of ourselves as a data company. We don't produce any drugs, we don't produce any therapies. We do genomic sequencing, as I'll show you in a moment, but a large part of what we do is collect clinical follow-up data. We structure data in about 60 to 70 fields coming from the medical record. The fields, as an oncologist, that you might expect, demographic information, histologic information, genomic information, outcome information, and so forth. And the thought is, by getting every patient's record into 60 to 70 structured fields, you can then put them all in the same database, along with the genomic information, and analyze it. And currently, I'm pretty sure we have the world's largest combined clinical and genomic database. What we're really expert at is structuring unstructured clinical data. We do it through a combination of optical character recognition, natural language processing. That can get us maybe 60, 70% of the way there, and then we have over 150 data analysts that make sure that that data is accurate. We structure all of the ASCO Cancer Link medical records, for example. Not all of them have sequencing data, but a lot of them do. So we're structuring now about 3,000 records a week. We'll do close to 200,000 records this year and throw that structured data into the same database for analysis. So we basically do a couple different things. We do sequencing. We have our go-to test is a 595 gene test. No magic about that number of genes. But what is different compared to some other sequencing companies is we do a normal blood sample in every case. We think that that's important to determine what's germline versus what's somatic. And we also do RNA expression profiling in every case. And we've already seen interesting patterns where, for instance, you can have RNA overexpression of EGFR, where we couldn't find any EGFR mutation at the DNA level. It might be there, but we couldn't find it. And it turns out the one case I'm thinking about responded to erlotinib in that particular situation. So we do normal blood, DNA, and RNA. But it's the clinical data structuring, as I say, that really, I think, sets us apart from anybody else. We've gotten to be very efficient at this. We can do it at a fairly reduced cost, fairly quickly. And when we give the result of the sequencing back to the doctor, we also give them the structured clinical information at the same time and keep a de-identified copy into our database. And we can talk maybe at the panel session some of the legal and regulatory issues around that. This is just an example of our particular sequencing test. The test itself is not that different other than what I mentioned. We'll give the genomic variants that we think are actionable. Those that are germline we'll also list. We'll do TMB, we'll do microsatellite instability. What does make us a bit unique is when we put down the clinical trials, we've also structured over 9,000 clinical trials in the US using the same criteria of 60 to 70 data fields. So we can match the patient to clinical trials at that level and not just report a trial because it happens to require EGFR mutation or it happens to require something else. Now, we can't match it down to every entry criteria, but the trials we list, there's going to be a much better chance that that patient is going to qualify for the particular trial that we do list. Here's an example of how we'll list, in a brief fashion for the doctor, the clinical story of the particular patient. So what this allows us to do is analyze a particular patient's genomic result compared to every other genomic patient in the database and every other patient that's clinically similar in the database as to what happened. What patient treatments did they get? What did they respond to? How many times did they respond? All of the information that then the doctor can look at and make at least a little bit more informed decision whether they should pull the trigger on a particular treatment. We do that in a number of ways. Actually, in our portal, each doctor gets what they call a cohort view, where their patient is in the middle. Each dot is another patient in our database that has the same histology, and they can filter the histologies. The closer they are to the center, the closer the genomic profile of that patient is to the patient in question. All of the treatments for that particular tumor type are listed here with the response rates. And as you manipulate this, you can see changes in the response rates. So if you only wanted to look at KRAS mutated pancreatic cancer, for example, as a clinician, you could put that in here and see if the response rates to Gemabrax are better than FOLFIRINOX, for example. Now, we know that clinicians are not going to generally have time to do this. It's available. They all get it with the portal if they order one of our tests. But we are also doing it internally. And we're coming up with what we call a series of insights that then we will put back on the report in a distilled fashion so the doctor gets it and doesn't have to do all this analysis him or herself. So I'll finish up by just showing you the kinds of insights that we can put on the report. We've been sequencing now for almost a year and a half, and we have enough data now that we are now starting to put insights back on the report. Not every patient is going to have an insight that we can say, it might be so rare a mutation or a tumor type that we just don't have enough cases yet to conclude anything. But we have enough in enough patients that we're going to start, in fact, we have started putting these back on the report. So, for example, I think the most significant one is the patterns of prescribing and outcomes, which I'll show in a moment. We can also just list the molecular frequencies, the numbers of pancreatic cancer patients that have this mutation. We also have come up with a disease classifier through the RNA expression that will predict, for example, in tumors of unknown origin, what the likely histology is based on all the other cases that we have. Same with prognostic indicators. We do pathway analysis, and then very interesting, I'll show you in a moment, where there's lack of concordance between RNA overexpression, immunohistochemistry. We'll highlight that and give some insights as well. So just quickly to go through these, here's a prescribing pattern outcome. So the insight here in colorectal adenocarcinoma, patients with the pathogenic variant of APC, which this patient has, show the following therapeutic incidence and response. And it turns out the response rate to 5-fluorouracil leucovorin and oxaliplatin seems to be 27% in the others you see here. So the point is the doctors get a sense as to what types of treatments patients have had and what kinds of responses there have been. Here's an example of the molecular frequencies. Just again, it's not as earth-shattering as the previous one, but we'll say here BRAF variant V600E was observed in only 1.2% of the pancreatic patients in our database. So the doctor isn't going to expect a whole lot of trial data out there. Disease classification. For all of the tumor of unknown origin cases that come in as TUO, we will give the predicted primary origin based on our RNA expression data listed here. For those patients that we have enough data, we'll give prognostic indicators. And we, again, since we have follow-up data, we don't just get the data when we sequence, we get data at every 6-month intervals from the doctors. We'll have progression-free survival data. And we can put that, generate that, and add it as an insight in particular situations. And then pathway analysis will pictorially indicate where the mutations are and where the drugs might work, that sort of thing. And then, as I mentioned, in situations where there's discrepancy between immunohistochemistry, for example, and DNA. I'm a breast oncologist by training, and HER2 increase is a perfect example of that. There are now 4 ways to look for HER2 overexpression. And the correlation is pretty good, but it's not 100%. So we've had cases where doctors have tested for HER2 overexpression in 4 different ways. Only 1 of them has been positive. 3 of them have been negative. And as a doctor, I can appreciate this. Most of the doctors would treat based on the 1 positive, figuring we're going to give the woman the benefit of the doubt that she may respond to Herceptin, even though the other 3 ways of measuring HER2 overexpression didn't show anything. So here we will point out that there is a discrepancy between IHC, which was positive, and our testing, which showed no evidence of amplification or overexpression. Hopefully we'll be able to lend some insight as to what the truth is. That's really what doctors want to know. But at least we'll point out the discrepancy at this point as we go forward. 2 other things in the last minute or so. We have the ability, again, since we're a data company, to give back to institutions all of their own data, which we do routinely. All of the raw sequencing data, all of the demographic data, all of the clinical data. And we give them a tool called Lens, where we can actually, they can actually filter all of their particular patients. You'd be amazed the number of prominent institutions when you ask them how many HER2-positive breast cancers have they treated in the last year. They don't know. There is no way to pull that data out. Or a pharma company will say, we're doing a study on IDH1 mutations. How many have you seen over the last year? They can't answer that question. It's easy to answer here. They just have to look at IDH1 mutations and glioblastomas, if that's what it is, and you can filter down and look at really any combination that you want to of your own data. And then lastly, I want to talk just a second about another thing that we're doing now, which is our Tempus R program. When I say we collect clinical data, most of the clinical data is from progress notes in the medical record. A very important piece of clinical data is radiologic data. As you know, the criteria for response, the RECIST criteria, have to do with certain measurements on CAT scans and x-rays. Well, most doctors don't do that. Most radiologists don't do that outside of a trial. We are getting in CAT scans, radiographs from about 20% of the patients that we deal with, and we can actually change these into 3D images. We can do volumetric analyses on these. We can do analyses of the density of the tumor. Again, the thought being that maybe there's some clinical change early on in the density of the tumor. Maybe looking at volumetric changes is going to be a more sensitive measure of early tumor response than biperpendicular diameter measurements. We don't know, but we're collecting all this data, again, as a data company, and we'll feed this into the database as well, and you can just imagine some of the interesting studies that we'll be able to do. So I'll just close with one of my favorite slides by William Osler. If it were not for the great variability among individuals, medicine might as well be a science, not an art. And by collecting as much data as we can and analyzing it, hopefully we'll give some insights to the genomic revolution in cancer. Thank you.